The novel coronavirus COVID-19 infiltrates human cells by means of a protein receptor called angiotensin converting enzyme, aka ACE2. To do this, the spike of the coronavirus, which is the S protein, binds to a spawn, the ACE2.
When the virus binds to ACE2 receptors with the help of another protein called serine protease, it makes its way into the cell, makes copies of itself, and spreads.
To understand the ACE2, we first have to look at the renin-angiotensin system,
which involves the kidneys, liver, lungs, blood vessels, and adrenals. First, the liver produces a protein hormone called angiotensinogen. When there is a drop in the blood pressure and thus narrowing the blood supply to the kidney secretes a chemical called renin into the bloodstream. Renin links to the existing angiotensin and converts it into angiotensin 1. Another enzyme is produced in the lungs called angiotensin converting enzyme, aka ACE, which links to angiotensin 1, converting it to angiotensin 2, the active configuration of the hormone. Angiotensin 2 then acts on both the peripheral arteries throughout the body and the adrenal glands, causing constriction of peripheral blood vessels and increasing sodium to raise blood pressure and restores blood flow back to the kidney. The angiotensin 2 further interacts with ACE2 protein and converts it into angiotensin 1,7, which acts as a vasodilator on the blood vessels and eliminating the action of angiotensin 2 on the adrenals. Angiotensin 1,7 is basically the counterbalance to the renin-angiotensin system.
This is how the renin-angiotensin system is related to the ACE2 membrane protein that the COVID-19 virus uses to begin the infection process. To develop therapeutic agents for the COVID-19 virus targeting the ACE2 receptors, there are medications that inhibit the renin-angiotensin system, which may prevent coronavirus infections and replication via the ACE2.
First is the angiotensin converting enzyme inhibitors, or ACEIs, which blocks angiotensin converting enzymes from the lungs from transforming angiotensin 1 to angiotensin 2. The second is the angiotensin receptor blockers, or ARBs, which blocks angiotensin 2 from binding to its receptors on the blood vessels and adrenal glands.
Instead of inhibiting the renin-angiotensin system, researchers may directly target the ACE2 receptors.
First, researchers may try and cover the spikes on the virus with antibodies to stop them from binding with the receptor without interfering with the effects of ACE2. Second, the binding of the coronavirus to ACE2 requires a protein called serine protease, and this protein can be inhibited by camostat mesylate and thus blocks the entry of the virus into the lung cells.
Although there is only limited clinical research information at this time, there is unlimited potential to broaden the spectrum of the therapeutic treatments for COVID-19.